Hey students, in this module, we'll be talking about comparing sequences from the biological databases. It's a very important module as it will lay the foundation for such an exercise wherein we compare the sequences from different sequence databases. You already know that there are a large number of sequence databases that are available online for you, and GeneBank and UniProt are two of them. What if I ask you that you take two sequences from one such database and compare them? For instance, if I ask you to take two protein sequences and compare them, what information can this comparison lead us to? One obvious thing is that it can tell you how similar these two sequences are. Or it can also tell you which specific differences exist between the two sequences. Moreover, it can tell you how the evolutionary process has evolved and how different speciation and differentiation events have occurred in the past. Moreover, it can also help you conclude the relationship between these two sequences. Now let's extend this example to the DNA and RNA sequences as well. You can draw the same conclusions by looking at the sequences from the DNAs as well as the RNAs. Let's consider this sequence and see how such a comparison can be done. So in this case, this is a DNA sequence, as you can see from the T's. So you can simply make it a rule of thumb that if a nucleotide sequence is given to you and it contains T, then it cannot be an RNA sequence. So this is a DNA sequence and I want you to compare it with another sequence. So let's consider another sequence A, C, T, G, and G. So one obvious difference is that this sequence is longer and this sequence is short, as it has only 5 nucleotides while this has a lot more than 5. Secondly, you can easily see that the nucleotide sequence that I mentioned here is exactly the same as the first 5 nucleotides in the first sequence. So this is another information that you can draw. So if you want to do a similar comparison with the RNAs, then you have to take an RNA sequence, and you can guess that by looking at these uracils here. And if I ask you to compare it with U, A, G, C, and U, so obviously this sequence is shorter again and this one is long, but you will see that this sequence matches this portion very nicely. Also, it is very similar to this sequence, but there are two differences here and here. So it matches as well, but to a lesser degree. The area of the sequence highlighted in red matched perfectly, while this portion matched partially. So you can have these very insightful search results by comparing different sequences. Let's consider an example from the proteins. So this sequence is a protein sequence. As you can see that it's composed of 20 different alphabets, probably, and does not limit to A, T, C, and G or A, U, C, and G. So this was the DNA sequence nucleotides. These are the RNA sequence nucleotides. So this is a protein sequence. So by looking at this protein sequence, if I ask you to compare this sequence, so you will say that probably this sequence is very close to this portion. However, we have three mismatches. So here LFT exists, here I exists. The rest of the sequence matches very nicely: M, Q, V, K, and L. But in this case, there are three amino acid mismatches. So these are the cases that can exist, and these can help you actually, as we will see later, to look at similarity between sequences, the specific differences, their evolution, as well as the relationship in between the sequences.